Imagine a protein inside your brain, right? And it's acting like this master mechanic, just frantically repairing damaged cancer cells so they can survive chemotherapy.
Yeah, wild.
But then imagine that exact same protein, if it somehow gets pushed outside the cell, it suddenly acts as a blaring fire alarm.
Right.
And that alarm literally calls in the immune system to destroy that very same cancer. Welcome to the deep dive.
Thanks for having me.
Today, we are unpacking this truly groundbreaking paper. It was published in July 2026 in the journal Cell Death and Disease. And, it answers a massive question in oncology.
It really does. The mission for this deep dive is to really understand this previously unrecognized cellular mechanism, because it drives one of the most aggressive cancers we know of.
Yeah.
We're going to look at how a tumor basically manipulates its own internal machinery to stay alive. And, more importantly, how researchers figured out a way to turn that sabotage into a weapon against the cancer itself.
Okay, let's unpack this. We have to start with the grim reality of the problem here, because if you've ever, looked into brain cancer, or if you know someone who's faced it, the landscape is incredibly difficult.
Definitely.
We're talking specifically about glioblastoma today, or GBM.
Right. And just to give some context for you listening, glioblastoma is the most aggressive primary brain cancer in adults.
Yeah.
The statistics are, well, they're sobering, honestly. Even with aggressive treatment, the median survival is only about 14 to 16 months.
Wow.
Yeah, and the 5-year survival rate sits below 5%.
That's just brutal. And the standard treatment protocol for a patient, it usually involves surgery, right? To remove as much of the tumor as safely possible.
Exactly.
Followed by radiation, and then this very specific chemotherapy drug called temozolomide, or TMZ for short.
Right, TMZ. It's an alkylating agent.
Okay, break that down for me. Which means what exactly in practical terms? Like, how is it actually killing the cancer cells?
Well, it works by fundamentally damaging the tumor cell's DNA. So when a patient takes TMZ, the drug travels to the brain and starts attaching these little chemical tags, specifically their methyl groups, and it attaches them to the DNA inside the tumor cells, targeting this very specific spot on the DNA ladder called the O6 position of guanine.
So it's essentially dropping like biochemical bombs on the cancer's genetic code.
That's a great way to picture it, yeah.
Yeah.
When the cancer cell tries to divide and replicate its DNA, the cellular machinery hits that damaged, methylated spot, and it basically derails.
Right.
The DNA strands break, the whole replication process collapses, and the cell is supposed to undergo apoptosis, which is programmed cell death.
Which sounds like a really solid strategy on paper.
It does.
But here is the massive clinical hurdle that this paper is addressing. It doesn't work for long.
No, it doesn't.
In over 90% of recurrent glioblastoma cases, the tumor develops resistance to TMZ. Like the chemotherapy just stops working entirely and the tumor starts growing again.
Right. And for years, oncologists and researchers have just been trying to figure out why. Multiple theories have been proposed.
Like what?
Well, maybe the cancer has specialized stem cells, or maybe the tumor builds these microscopic pumps to literally spit the drug back out.
Oh, wow.
Yeah, but the complete overarching molecular network that coordinates this resistance, it's remained a mystery. Up until now, anyway.
And this brings us to the primary villain of our story today. It's this protein called E2F6.
Yes.
E2F6.
Now, in a healthy cell, what is E2F6 supposed to be doing normally?
So normally E2F6 is a transcription factor. If you picture your genome as this massive library of instruction manuals, transcription factors are basically the librarians.
Okay, I like that.
They physically bind to the DNA and decide which books get opened and read and which ones get locked away. And E2F6 specifically acts as a repressor, meaning it turns genes off.
Okay. But in glioblastoma, the researchers found that this librarian is completely out of control, right? It's severely overproduced. Exactly. And what's fascinating here is the sheer scale of the clinical data they used to prove this.
Yeah, they didn't mess around.
No, they didn't just look at a few isolated cases in a lab. They analyzed massive genomic databases. Specifically, the Cancer Genome Atlas and the Chinese Glioma Genome Atlas.
Those are huge.
Right. These databases hold genetic information from thousands of patient tumor samples.
And so what did that massive data set actually show them?
It showed a very clear, very dark pattern, honestly. They found that E2F6 levels shoot up significantly as the tumor gets deadlier.
Okay.
Brain tumors are graded by the World Health Organization from grade 1 to grade 4. Grade 2 is, diffuse and slow growing. But grade 4 is glioblastoma.
The worst one.
Yeah, highly aggressive, necrotic, highly vascular. And the researchers saw that as you move from grade 2 up to grade 4, E2F6 levels just skyrocket.
So the higher the E2F6, the higher the recurrence rate, and the worse the overall survival for the patient. I read that the tumor tissues literally stain darker for this protein under a microscope compared to normal brain tissue.
They do, yeah.
So E2F6 is like a corrupt factory manager in the tumor. It's changing all the standard operating procedures to keep the factory running despite the chemotherapy bombs dropping from above.
That's a perfect analogy.
But I have to admit, I'm a little confused here. Just knowing E2F6 is high, that doesn't really explain the actual mechanics of it all.
True.
Like, how does this corrupt manager physically stop the TMZ chemo from working? What is it actually doing inside the nucleus?
Well, that is exactly the question the researchers had to answer next. To find out what E2F6 was doing, they had to use some incredibly advanced sequencing technologies.
Okay.
First, they used RNA sequencing to see which genes were being turned on or off when they artificially removed E2F6. Then they used this cutting-edge technique called CUT and tag.
CUT and tag. Okay, if you're listening and wondering how researchers actually track this stuff, CUT and tag sounds like some sort of molecular tracking device.
Yeah.
How does that actually work?
It basically is a tracking device. CUT and tag stands for cleavage under targets and tagmentation.
Wow. That's
Yeah,
okay.
quite the name. But imagine you want to know exactly where our corrupt manager, E2F6, is standing inside that massive library of the genome.
Right?
Researchers use a specific antibody that acts like a bloodhound. It's designed to only hunt down E2F6. And attached to that bloodhound is a little enzyme.
So when the bloodhound finally finds the E2F6 protein...
The attached enzyme essentially takes a molecular photograph.
Oh, that's cool.
Right. It cuts the DNA right at that exact spot and attaches a unique barcode tag to it.
I see.
So by collecting all those tags, researchers can map the exact physical locations where E2F6 is binding across the entire genome.
That is just incredible technology. So they overlay that broad RNA data with this highly specific CUT and tag map. And what do they find?
They found a massive binding peak right at the promoter region of a gene called SH3GL2.
Okay, the promoter region.
Yeah, the promoter is essentially the on switch for a gene. E2F6 was directly binding to the SH3GL2 switch, completely locking it down and shutting off its production.
Okay, let's process this for a second. The tumor panics because of the chemotherapy, so it pumps out this corrupt manager, E2F6.
Yes.
And the very first thing E2F6 does is sprint over and shut down the SH3GL2 gene.
Exactly.
So why is the tumor so desperate to turn that specific gene off? Like, what does SH3GL2 actually do?
Well, in a normal, healthy context, SH3GL2 is a membrane remodeling protein. Cells need to move things in and out constantly, right?
Right.
SH3GL2 is involved in bending the cell's outer membrane to form these little transport bubbles. They're called vesicles.
Okay.
And these vesicles securely package molecules to be shipped outside the cell.
Got it. So it's basically the cell's shipping and receiving department.
Exactly. But its role in chemotherapy resistance was completely unknown before this paper.
Wow, really?
Yeah. So to figure it out, the researchers ran a mass spectrometry analysis. They basically cast this wide chemical net to catch any other proteins that SH3GL2 interacts with.
And what did they catch?
They found it teams up with a large cellular shipping complex called ESCRT3. And specifically, it binds to a protein in that complex called CHMP4B.
Okay, I have to ask about the structure here, because the paper goes into heavy detail about how these proteins physically connect to each other.
It does.
How does SH3GL2 actually grab hold of this shipping complex?
This is where the structural elegance of biology just really shines. SH3GL2 has two distinct biological hands, basically, known as domains.
Okay, two hands.
Right. On one side, it has what's called a bi-R domain. On the other side, it has an SH3 domain. And the researchers actually mutated these hands one by one in the lab just to see what would happen.
Oh, clever.
Yeah. They found that SH3GL2 uses the bi-R domain hand to firmly grab onto that CHMP4B shipping complex we talked about.
Okay, so it uses one hand to hold the shipping container. What is it doing with the other hand?
Well, it uses its other hand, the SH3 domain, to grab onto a very specific payload, a protein called HMGB1.
And there it is. This is the protein from our hook at the beginning of the deep dive, the mechanic that doubles as a fire alarm.
Precisely. HMGB1 is the absolute crux of this entire resistance mechanism.
Okay.
When a healthy cell is stressed by a toxic drug like TMZ, SH3GL2 is supposed to step in. It grabs the shipping complex with one hand, grabs HMGB1 with the other hand, packs the HMGB1 into a transport vesicle, and physically ejects it out of the cell.
Let me just make sure I have this chain reaction right, because this is devious.
It really is.
The chemotherapy, TMZ, enters the brain. The glioblastoma tumor senses the danger. It cranks up the corrupt manager, E2F6.
Yep.
E2F6 goes straight to the DNA and shuts down SH3GL2. Because HMGB1 is gone, the shipping department shuts down. It can't grab the HMGB1, which means HMGB1 is never ejected. It gets trapped inside the cancer cell.
That is exactly the pathway. You nailed it.
So why does it matter if HMGB1 is trapped inside? Like, what is it actually doing in there?
To understand that, we have to look strictly inside the nucleus of the cell. HMGB1 stands for High Mobility Group Box 1.
Catchy.
Right. But it is a highly conserved master DNA mechanic. It naturally binds to chromatin, which is just the bundled up form of DNA.
Okay.
Now, remember how we said TMZ works? By causing double-strand breaks in the DNA?
Right, dropping those biochemical bombs to derail the cell's replication?
Well, when those DNA strands break, any HMGB1 that is trapped inside the nucleus immediately rushes over to the damaged sites.
Oh, wow.
Yeah, it acts as an emergency scaffold. It physically binds to the broken DNA ends and facilitates the rapid recruitment of a massive repair crew.
So it's literally calling in the biological emergency responders to fix the damage the chemo just caused.
Yes, it activates two major DNA repair pathways. The first one is homologous recombination, or HR. This is a highly precise pathway where the cell uses a flawless copy of the DNA as a template to perfectly fix the break.
And the second pathway?
That would be non-homologous end joining, or NHEJ. This one is much faster, but it's sloppier.
Sloppier how?
It doesn't use a template.
Yeah.
It essentially just grabs the two broken DNA ends and duct tapes them back together.
Okay, I see.
It's prone to errors, sure, but it keeps the cell alive. And HMGB1 supercharges both of these repair processes.
This tumor is incredibly smart. By keeping E2F6 high, the tumor ensures the shipping department is closed, trapping all the HMGB1 master mechanics inside the building.
Exactly.
So no matter how much chemotherapy you drop on the tumor, the trapped mechanics just constantly rebuild the damaged DNA. No wonder the chemo stops working.
It's a brilliant survival strategy by the cancer. And the researchers proved this beautifully in the lab using well-known glioblastoma cell lines, specifically U87 and U251 cells.
Okay.
They wanted to see what happens if you break the tumor strategy. So they artificially knocked down the corrupt manager, E2F6, using targeted RNA interference.
So they essentially fired the corrupt manager. What happened?
Well, as predicted, when E2F6 dropped, the SH3GL2 gene was no longer locked down. Its levels bounced right back up. The shipping department came back online, grabbed the HMGB1 mechanics, and kicked them right out of the cell.
And without their mechanics inside, the tumor cells completely lost their ability to repair the chemotherapy damage.
Completely. And we know this because the researchers visualized it using something called a COMET assay.
Oh, a COMET assay. I've seen pictures of these. They really do look like little shooting stars under a microscope. How does that assay actually show DNA damage, though?
It's a really clever technique. You take the individual cells and place them in a specialized gel. Then you apply an electrical field across the gel.
Okay.
DNA has a natural negative charge, so it wants to move toward the positive electrical pole. Now, if the cell's DNA is intact and healthy, it's a massive tangled ball that is just too big to move through the thick gel. It stays in one round spot.
The head of the comet.
Right, exactly. But if the DNA is heavily damaged and fragmented into thousands of tiny broken pieces by the chemotherapy, those tiny pieces easily get dragged through the gel by the electrical current.
Ah, I get it.
Yeah, they pull away from the main cell body, forming a long tail.
So the longer the comet tail, the more shattered the DNA is.
Exactly. And when they knocked down E2F6 and added the TMZ chemo, the glioblastoma cells showed massive comet tails.
Wow.
They also saw a huge spike in a marker called gamma H2AX.
What's that?
It's basically the universal cellular distress signal for severe DNA double-strand breaks. So the repair factory completely collapsed, the cancer cells died, and the chemotherapy worked again.
That alone is a massive breakthrough. But this raises a huge question for me, and it takes us right back to the hook of this deep dive.
Okay.
In a scenario where we successfully knock down E2F6 and the shipping department kicks HMGB1 out of the cell into the void, what happens to it? Like, does it just float away like biological garbage?
This is where the story shifts from cellular mechanics to full-blown immunology.
Okay.
Outside the cell, HMGB1 is not garbage at all. It takes on a completely different identity.
Extracellular HMGB1 acts as a prototypical DAMP.
A DAMP, like DAMP. Yes. A damage-associated molecular pattern. Think of it as a blaring biological fire alarm.
Okay, so the cell ejects this protein, and suddenly it's screaming that something is wrong. Who is it screaming at?
The body's immune system. Specifically, we need to look at the tumor microenvironment.
The neighborhood.
Exactly, the neighborhood immediately surrounding the cancer. Tumors are incredibly hostile neighborhoods. They survive by brainwashing the local immune cells, particularly cells called macrophages.
Macrophages are usually like the Pac-Man cells that eat debris and foreign invaders, right?
Normally, yes, that's their job. But tumors secrete chemical signals that force these macrophages into what's called an M2 state.
Okay, M2.
In the M2 state, the macrophage is essentially hypnotized. Instead of attacking, it actively protects the tumor, it suppresses other immune cells, and it even helps the tumor grow new blood vessels.
So the tumor basically has its own private, brainwashed security force.
Exactly. But when that HMGB1 fire alarm is suddenly ejected into the neighborhood, it binds to specialized receptors on those macrophages, completely overriding the tumor's brainwashing.
Oh, wow.
Yeah. The researchers proved this by taking the liquid surrounding cells that were releasing HMGB1, called conditioned medium, and exposing it to those brainwashed macrophages.
And what did the fire alarm do to them?
It forced the macrophages to polarize into an M1 state.
And the M1 state being the aggressive anti-tumor attackers.
Precisely. They wake up from the hypnosis, they stop protecting the tumor, and they start pumping out massive amounts of inflammatory signals.
Like what kind of signals?
They release cytokines like TNF-alpha, interleukin-1 beta, and interleukin-6. The neighborhood goes from this quiet, immunosuppressed safe haven for the tumor to a raging hostile war zone.
That's incredible. But, testing this in a petri dish with conditioned medium is one thing.
Sure.
The human brain is infinitely more complex. Did the researchers prove this fire alarm actually works inside a living, breathing immune system?
They did, and the methodology they used here is crucial. They didn't use standard laboratory mice that have their immune systems wiped out.
Which is common, right?
Yeah, it's very common in cancer research just to get human tumors to grow in the mice. But to test a fire alarm, you need a fire department.
Right.
So they used an immunocompetent GL261 mouse model.
Meaning these mice had fully functioning, healthy immune systems.
Correct. They implanted the glioblastoma tumors into the brains of these mice, let them establish themselves, and then treated them with the TMZ chemotherapy.
And when they manipulated the tumor to lower E2F6 and release the HMGB1 fire alarm,
the results were just stunning. Not only did the tumor shrink from the TMZ because they couldn't repair their DNA, but the researchers saw a massive remodeling of the immune environment.
How could they see that?
They used a technique called multiplex immunofluorescence. It allows you to tag different types of immune cells with different glowing colors and look at them all at once under a microscope.
That sounds like a cool image. So what colors lit up the neighborhood?
They saw a huge influx of those aggressive, awakened M1 macrophages. But the fire alarm didn't just stop there. No, it brought in the heavy artillery from the adaptive immune system. They saw elevated infiltration of CD8-positive T cells.
The executioners.
Yes, these are the specialized executioners of the immune system that directly kill cancer cells.
Yeah.
They also saw an influx of natural killer, or NK cells.
So the whole cavalry arrived.
Exactly.
Did the tumor's original security force, the immunosuppressive cells, did they stick around?
No, they didn't. At the exact same time the attackers flooded in, the immunosuppressing regulatory T cells, the TREGs, they significantly dropped.
Wow.
The tumors shrank dramatically, and the mice with the E2F6 knockdown had significantly prolonged survival compared to the controls.
I am just marveling at the efficiency of this 2-for-1 mechanism. I mean, let's think about what this means practically for you listening. If you can target this corrupt manager, E2F6, you don't just make the tumor unable to fix itself. The sheer act of the tumor failing to fix itself actively calls in an immune system airstrike on the wound cancer. You strip the tumor of its armor, and you hand a GPS locator to the immune system, all in a single biochemical move.
It is a remarkable finding. I mean, we have known for decades that TMZ resistance is the biggest roadblock in treating glioblastoma.
Yeah.
What this paper does is map out a brand new, previously unrecognized mechanism linking a cancer cell's internal DNA repair capacity directly to its external tumor immune regulation. Everything hinges on this E2F6 to SH3GL2 to HMGB1 axis.
So to bring this all together, we started by talking about the murky landscape of oncology and how glioblastoma has managed to evade our best weapons.
Right.
What this Cell Death and Disease paper provides is a new molecular blueprint. It illuminates the hidden machinery, the corrupt managers and the trapped mechanics that make this brain cancer so deadly.
Exactly.
Targeting this pathway, perhaps by finding new drugs that can specifically inhibit E2F6 or maybe artificially boost SH3GL2, that could be a massive leap forward.
It really could.
It could restore the power of chemotherapy for patients who desperately need it, while simultaneously turning their own immune systems against the tumor.
And, as we close, this research raises an important, broader evolutionary question for us to just kind of chew on.
Oh, I love these. Let's hear it.
Well, evolution is incredibly economical, right? Isn't it fascinating that biology uses the exact same molecule, HMGB1, as a vital tool for survival and repair on the inside of the cell and as a blazing signal for destruction on the outside?
That is fascinating.
It really makes you wonder how many other mechanic proteins are hiding inside our bodies, harboring secret second lives, just waiting to be discovered by the next generation of researchers.
A secret double-agent protein that could drive the future of cancer treatment. It's a perfect reminder of how much is still left to uncover. Thank you for joining us on this deep dive into the source material. Keep questioning the consensus, keep looking at the data, and most importantly, stay curious. We'll catch you next time.